Hello and welcome to Pitch Talk Session 3. We hope you are ready to learn something new and ask that you vote for your favorite presentation by following the link at the end of the video. First up, we got Human Azizi from McGill University.
Many of you are familiar with various psychiatric disorders, such as schizophrenia and obsessive-compulsive disorder or OCD. Although there has been extensive research on each of these disorders, they are still not well understood as they are complex and polygenic disorders. As a result, there is still no definitive treatment for them, and physicians currently rely on deep brain stimulation or pharmacological methods to improve the conditions of these patients. Unfortunately, these methods lack specificity as they manipulate broad regions of the brain or entire neurotransmitter systems, leading to various off-target effects. Recent technological advancements have allowed us to tackle these problems from a new perspective. Using all optical approaches, we can combine optogenetics and calcium imaging to manipulate the activity of specific neural subpopulations while simultaneously measuring the activity of other neural populations. Since the specific psychiatric disease symptoms reflect changes in the basal activity or synaptic plasticity in certain neural circuits, we can use this all-optical approach to identify relevant circuit abnormalities in animal models of psychiatric disorders or generate new circuit models. In my research, we examined the role of basal ganglia dysfunction in compulsive behaviors that are typical of OCD and Tourette syndrome. Using an all-optical approach, we focused on the nucleus accumbens and two glutamatergic inputs to the accumbens originating in the hippocampus and paraventricular thalamus. In human patient populations, dysregulations at various junctions in this circuitry have been implicated in the presentation and severity of compulsive behavior. So we initially showed that repeated activation of these pathways elicit compulsive grooming and cognitive inflexibility on a reversal learning task. So we focused on these pathways for our all-optical investigations. In the next phase of my research, and as a proof of concept, we activated hippocampal neurons using optogenetics and recorded calcium transients from downstream nucleus accumbens neurons. Although we were able to evoke population responses, we found that repeated stimulations of accumbens projecting hippocampal neurons didn't affect the size of these evoked responses. We then decided to look at the natural responses of these neurons instead. So we took advantage of a well-established phenomenon that nucleus accumbens neurons activate at the onset of locomotion. We isolated these instances and found that endogenous activity of these neurons at the onset of locomotion was heightened after repeated hippocampal neuron stimulation, suggesting that this pathway was in fact potentiated by our optical manipulations. So overall, my research shows that these pathways are implicated in compulsive behaviors and that we can monitor changes in pathway function using novel all-optical approaches. In future, I plan to apply all-optical techniques to other input pathways and synaptic junctions relevant to compulsive behaviors. Thank you.
Thank you, Human. Next up, we have Gabriel Blanco Gomez from McGill University.
In April of 1861, a 50-year-old man was admitted to a hospital in France. Let's call him Mr. X. At first glance, Mr. X looked fine. He had no signs of stroke, he could walk, he could smile, but there was one thing that he couldn't do. He couldn't speak. Mr. X was going to die in the next couple of weeks, but his case wasn't unique. You see, at the time, there was a young neuroscientist by the name of Paul Broca who was doing autopsies on patients like Mr. X. He was interested in figuring out what was going on in the brain of these patients that was causing language deficits. And then one day he found it. He found a small region in the left frontal lobe that he called the language area. At the time, it was a major breakthrough. He was able to show that the brain had different sections or regions, and that each region had a particular function. But he also noticed something else, something interesting. All of these patients had damage only to the left side. But why? I mean, if you look at the brain, it has two hemispheres, a right and a left hemisphere. So why is language on the left? This is the question that I want to answer in my research. I want to know how the brain develops lateralization and what happens when that system fails. To answer that question, I work with children at risk for ASD, or autism spectrum disorder. What we do is we bring these cute little babies to the lab, with the parents' permission, of course. We have them put on an EEG mask or an EEG cap, and we basically measure how much or which hemispheres are dominant during which task. And what we found so far is remarkable. At 6 months of age, a child or an infant is already able to have a left hemisphere dominance when it comes to language. In ASD, however, there seems to be no lateralization at all. I like to explain this as a tandem bike. You see, in a tandem bike, you have two people who have to pedal for it to work. However, there's always one person at the front who decides when to go, where to go, and when to brake or cause inhibition. In ASD, it's almost as if you had two separate steering wheels with two separate brake systems, or two hemispheres that are fighting for dominance or resources when it comes to certain tasks. If you look at language, children that have less lateralization go on to develop worse language outcomes, which again is not surprised considering how important lateralization is for language. But it does lead to more questions. If these changes are happening at 6 months old, that means there has to be some type of genetic component to it. So can we find it? Paul Broca made a major scientific breakthrough 200 years ago, but he didn't have access to genetics, or at least not the way that we do. So I'm excited to find out what the next mysteries are that we can uncover.
Thank you for presenting with us, Gabrielle. Next up, we have Brianna Owusu from McMaster University.
I want you to listen to your heartbeat. Is it beating in a predictable rhythm or an unpredictable rhythm? Have you ever noticed that your heart can beat to the same rhythm of a tune? Our ability to synchronize to the beat of a song is called entrainment, and the same effect can be seen across our entire bodies. Imagine you're listening to a really groovy tune, and then all of a sudden, your feet start tapping, your fingers are snapping, and you might even start dancing in time to the beat of the music. This same synchronization can even be seen in the rhythms of our brains. Brainwaves can naturally synchronize to regular patterns that we hear, but for every 1 in 10 children that's diagnosed with dyslexia, the inability to read, write, and spell fluently, these synchronizations are not seen. In my PhD, I want to help find a solution for early interventions for at-risk kids of dyslexia, but I want a solution that's inclusive and diverse for a condition like dyslexia where reading is a lifelong challenge. And what if musical rhythms can help make a difference? Well, it turns out empirically that rhythms can help people with dyslexia, specifically rhythms that have a regular, predictable beat. When children hear predictable beats, their brains are primed to anticipate and predict likely words that could come up next in a sentence. But whether rhythm training can improve memory for unfamiliar words has never been thoroughly investigated. So in the world's first behavioral online study of its kind, I will be recruiting children with dyslexia and typical reading children to actively listen to rhythms that either have a predictable beat or an unpredictable beat. My research team and I will then measure their ability to anticipate likely words and sentences after hearing those rhythms. And we will also measure their ability to memorize made-up words that cannot be anticipated. But why word memory? Well, because people with dyslexia can also struggle with repeating and remembering words that they've just heard. This comes from a problem in the brain that's responsible for storing, rehearsing, and repeating words in memory. If shown effective, musical rhythms can be implemented in early interventions for dyslexia even before reading instruction begins. This means that children will not have to suffer the dire consequences of the currently practiced waiting to fail approach. This research joins the growing chorus of uncovering the heart of dyslexia. Thank you.
Thank you for your talk, Brianna. Next up, we have Bryce Robbie from Polytechnique Montréal.
Hello everyone, I'm Bryce Robbie, and I'm going to present you some research done at Jean Provost's lab at Polytechnique Montréal about deep learning for ultrasound localization microscopy in 3 dimensions. First of all, You should know that the goal of ultrasound localization microscopy is to map the blood network at high resolution. How is this done? The detection and precise localization of microbubbles injected into the bloodstream is used to deduce the position of blood vessels in ultrasound images. However, this requires a relatively long acquisition time, around 10 minutes, since it is necessary to wait until the microbubbles have covered the entire blood network. To reduce this acquisition time, we can increase the concentration of microbubbles. However, conventional detection methods are based on the assumption of isolated bubbles, and a high concentration of microbubbles invalidates this assumption. This leads to a degradation of the detection performance. In order to better model the interaction between microbubbles and thus to look at them with precision, even in high concentration, a deep learning approach has been developed in the laboratory. Based on realistic simulations, we train a neural network to predict the position of bubbles in a microfilm. The use of the microfilms allows the network to rely on a temporal context and thus to use the trajectory of the bubbles to better understand their interactions. Unfortunately, this approach requires a large amount of graphic memory and is therefore limited to 2D imaging for the moment. That's why it was necessary for us to develop a more memory-efficient deep learning model. For this purpose, the use of temporal context was concentrated in the trajectory detection part, where it is really needed. The deepest layer of the network uses a temporally flattened representation of the microfilm, which significantly reduces memory costs and allows for three-dimensional expansion. In the first step, we compared the results of the lightweight approach and the 2D greedy approach on a high concentration simulation set. The dice coefficient measures the precision of the predicted trajectory. So as you can see, the lightweight approach obtains similar performances to the greedy approach while reducing the memory consumption. Finally, we could extend the lightweight model to 3D. You can see in silico results in 3D and at high concentration of the lightweight and of the conventional approach. In these settings, the deep learning approach clearly outperforms the conventional one. Future work will compare this deep learning approach with the conventional one in vivo. Thank you for your attention.
Thank you for sharing your work, Bryce. Next up, we have Dasha Sandra from McGill University.
Imagine you hurt your back and you are choosing which doctor to go to. You have two options. The first one would prescribe you a standard painkiller that they would prescribe to anybody else in this situation. And the second one, before prescribing you the painkiller, would take a couple of physiological tests to find which painkiller would be best for your profile. So this is the whole premise of precision medicine, which is a new branch of healthcare using biological and genetic markers to find underlying patient profiles and tailor treatments to these profiles to make them more effective. And of course, precision medicine has shown some early successes in increasing patient survival for oncology patients and decreasing side effects for cardiovascular medication dosing. Now, patients have various reasons to believe that precision treatments are more effective. They're more expensive, they're more elaborate, and they require more attention from the practitioner. But more importantly, precision treatments are tailored to one's genetics, which is often believed to be the unique and defining quality about our underlying nature. And so we decided to test whether just believing that your treatment is tailored to your characteristics increases your treatment response in the context of pain. So we recruited 17 participants to participate in a study focusing on different factors of pain perception. They completed several sham tests, ostensibly to see if they were eligible for the study, and then were separated into two groups, the experimental and control group. Those in the control group just received more information about different kinds of painkillers and then given a standard placebo device to use for the pain task. Those in the experimental condition, however, received false test results from the tests that they have just completed and had their placebo device tailored to their individual profile based on these tests, and then received this device to use for the pain task. Then all participants completed a pain task where they rated how much pain they experienced while using the machine turned on or the machine turned off. And so we found some positive trend towards significance, with participants in the experimental group experiencing more pain relief than those in the control group with the standard device. However, our results are non-significant due to a very small sample size, and we're now trying to replicate these findings in a larger sample. We hope that this study and these results, once replicated, could shine a light on the role of expectations in the context of precision medicine.
Thank you to all the presenters. This session is over, and we ask the audience to please use the link to vote.